Assalamu alaikum dear students, I hope you all are fine and in the best conditions of your health. So in today's lecture we shall discuss about drug discovery and structure-activity relationship. So this is actually a preview lecture and we will revise the concepts which are involved in drug discovery and structure-activity relationships, right? So the key concepts which we will discuss in this lecture are: what do we mean by hit and lead? What is the Lipinski's rule of 5? What are different methods for drug discovery? And what are different stages of drug discovery? What do we mean by SAR? And what are the different physicochemical factors for optimizing the biological action of drug? Then we shall see about the Eason-Stedman hypothesis, and we will also see about what are different analogs approach for structure activity relationship, right? So as we know, the drug is any chemical substance that can have effect on a biological system, right? So when we are considering the drug discovery, when we are searching for new drugs, the first step is to identify a hit, right? So what's a hit? Hit is any chemical compound that exhibits certain desired biological activity during the screening process. Right? So when you are carrying out the screening process of many of compounds, and among those many compounds you get one compound that has certain desired biological activity, then this compound is called a hit, right? And a hit should reproduce the activity upon retesting. Means that if you have a compound, it tests positive for antibacterial activity, and if you test it again, it should again test positive for that activity. Right? So it is necessary that a hit must show reproducible biological activity upon retesting. Even if someone else at some other place in the world, if he is also testing that biological activity, that activity should come positive. Right? So this is called a hit. So once you get a hit, then you optimize the hit, because we want to optimize the hit to improve its physicochemical properties. Right? It should have improved pharmacological properties. Pharmacological properties means biological activity. It must have improved the toxicological profile and the pharmacokinetic properties, right? So once we have optimized these properties of hit, then hit becomes a lead compound, right? So once you get a hit, you refine it and you make a lead from that, right? So when we are searching for a hit and we are searching for a lead compound, then the use of combinatorial chemistry and high throughput screening play a very important role, right? What's combinatorial chemistry? So combinatorial chemistry is a method by which we can synthesize a large number of related compounds in just one time, right? So in single time, in single run, you can synthesize thousands of compounds, but thing is that those compounds should be related. For example, we can synthesize a number of esters in one run. You can synthesize 1000 esters in a single run by means of combinatorial chemistry. Then we have high throughput screening. So high throughput screening allows analysis and screening of tens of thousands of compounds at a single time. Right? So in just one run, you can analyze thousands of compounds for certain biological activity or their chemical characterization, right? So by means of combinatorial chemistry and by means of high throughput screening, so you can make things to work very efficiently. Now we have Lipinski's rule of 5. So Lipinski's rule of 5 is also known as Pfizer's rule of 5, or simply the rule of 5 or R5, right? So whenever you are considering to design a drug and you want to know whether the molecule you are designing, will it be able to show some biological activity, then according to Lipinski's rule of 5, if a molecule have a molecular weight of 500 or less, the C log P value is less than 5, the hydrogen bond donors are less than 5, and the hydrogen bond acceptors are less than 10, and there are no more than 3 rotatable bonds. So if a molecule have all of these parameters, then it will show Lipinski's rule of 5. So it will show the potential to act as a drug. So this is called Lipinski's rule of 5, right? So molecular weight of a drug must be less than 500. C log P value is actually indicator of the lipophilic profile. What's lipophilic profile? Lipophilic profile is the ability of a drug to get soluble in non-polar solvent, right? So C log P value should be less than 5 means the drug must have some polar effects in it, right? So if C log P value is greater than 5, then molecule becomes non-polar, right? So molecule must be polar enough so that it can cross through the blood system, because blood has water in it. So molecule must be somewhat hydrophilic so that it can dissolve in water, and it must have a lipophilic profile as well so that it can cross the cell membrane as well, right? So C log P value should be less than 5, should not be more than 5, and the hydrogen bond donors must be fewer than 5, and the hydrogen bond acceptors should be fewer than 10, because if there are a large number of hydrogen bond donors and the large number of hydrogen bond acceptors, then your drug will be very, very polar. So if a drug is very polar, it becomes difficult for the drug to pass through the lipophilic cell membrane. Lastly, the bonds should not be rotatable. The rotatable bonds should not be more than 3. Why? Because if the number of bonds becomes greater, if the number of rotatable bonds become greater, then that case molecules become very, very flexible, and there are large number of conformations possible for the structure. So with a large number of conformations that are available, then the molecule will show affinity for large number of receptors, which is not desired, right? So we want a molecule to be somewhat rigid so that it cannot show interaction with every sort of receptor. So in order to make the molecule rigid, so we have the restriction that the number of rotatable bonds should not be more than 3. So this is called Lipinski's rule of 5. Then we have the methods of lead discovery, right? So when you are discovering a lead, then there are 6 methods to discover a lead. One is the random screening. So we carry out random screening when we do nothing about the receptor target, right? So when you don't know anything about the receptor target, then this is random screening. So whatever you have in hand, you just check it for all possible biological activities, right? So when whatever structures we have, you test the structure for all possible biological activities. So maybe we get some biological activity which is desired, right? Next is serendipity. Serendipity is also called by chance discovery or the accidental discovery. So serendipity means that if you are looking for something but you end up searching for something else. For example, let's say that I'm searching for an antibacterial agent, right? So I'm searching for an antibacterial activity, but when I search for a compound, it doesn't show antibacterial activity, but it shows some antifungal activity, right? So I was searching for antibacterial activity, but I found out the antifungal activity for that drug. So this is called serendipity, accidental discovery. I was not expecting this, but I get this, right? This is called serendipity. So majority of the drugs, they are found by serendipity. Then we have molecular modeling. So molecular modeling is a computational tool. So in this way, what we do is that we have a ligand, we have a structure, and we bind the structure with the receptor molecule, right? And this is all done on the computer. So the molecular modeling is also called docking, right? So we dock the ligand onto the receptor, and we see whether the ligand is able to show affinity for the receptor or not, right? So in this way, this is called molecular modeling, right? Next we have is molecular manipulation. So molecular manipulation means, suppose that you have a medicine that's already available in market, but this medicine has some side effects associated with it. So what we do is that we take this medicine and we make some derivatives of this medicine, right? We make some analogs of this medicine. So what we do with the analogs, the derivatives of this medicine, they are checked for their biological activity. So it's a chance that one or more of the derivatives of this medicine are more effective and have lesser side effect than the parent drug. So this method is called molecular manipulation, in which we take the medicine which is already available in the market and we carry out some derivatives of this drug. We make some derivatives of this drug and see whether any of the derivatives have some more biological activity than the parent drug and has some lesser side effects or not. Next thing is molecular designing. So molecular designing is also a computational tool. Right? It involves designing a compound to fulfill a specific biological activity. So in molecular designing, what we do is that we take the receptor. We know the three-dimensional structure of the receptor, and what we do is that we join different fragments with the receptors. We see what fragments are able to bind with the receptor sites, and in the next step we join these fragments together to form a structure. Right? So molecular designing is also called the fragment-based drug design. Right? So by this method, by just considering the structure of your receptor molecule, you can make a structure of a drug that will be able to bind with the receptor. Right? This is called molecular designing, is also a computational tool. Right? Next is the metabolites of the drug. Right? So the basic theme of the metabolites of the drug is that the drug is metabolized inside the body. Right? So when the drug is metabolized, we get the different metabolites of the drugs, and it is possible that those metabolites of the drug may have some better biological activity than the parent drug as well. Right? So what we do is that once the drug is metabolized inside your body, we identify the metabolites and we check the biological activities of those metabolites as well. So it is possible that one or more of the metabolites are more potent or are more effective than your parent drug. Right? So this method is called the metabolites of a drug method. So after the search of lead discovery, let's consider what are the different stages of drug discovery. Right? The first stage of the drug discovery is to identify the biological target. Right? The biological target is the molecule, is the biomolecule that was previously functioning for the normal functioning of the body, but because of certain disorder, because of certain disease, this molecule is not working now. Right? So once your biological target, once your biomolecule stops working properly, then what happens, that body is not able to perform the normal body functions. Right? So first step in the drug discovery is that we have to identify the target. What should be the target with which our drug should act. Right? So the targets are normally three. Number one, there may be some enzyme, there may be some receptor, or there may be nucleic acids. Right? So although nucleic acids are very rare targets, but they are simple, they are very important targets as well. Right? So there are basically three targets that are important, that are considered in the medicinal chemistry. They are enzymes, receptors, and the nucleic acids. Right? So once we have identified the biological target, the next step is to confirm whether this is actually our target or not. Right? So it may be possible that you consider some as a target, a molecule as a target, but this was not actually your target. Right? So we have to confirm whether this receptor, this enzyme, or this nucleic acid is your target or not, because if sometimes it happens the drug may interact with more than one target, then in that case some side effects are produced. Right? So in order to make sure that this is your target, it is very important to confirm whether you have identified the target, whether the target is actually the target or not. So after validation of the target, next thing is to search for the lead. Right? So we have to search for the lead structure. So in order to search for the lead structure, we have 6 methods which have been described in the previous slide. So you can discover the lead by means of serendipity, by means of random screening, by means of molecular manipulation, by molecular designing, by metabolite studies, etc. Right? So once you have searched for the lead, once you have got lead, then you have to optimize the lead. Right? So in order to optimize the lead, we make use of structure-based drug designs, we make use of quantitative structure-activity relationship, and we make use of computer-aided drug design. Right? So basically the structure-based drug design is a type of computer-based drug design. So whenever you make use of computers for predicting the drug designing or for optimizing the lead structure, then this is considered as computer-aided drug design. So structure-based drug design is also a type of computer-assisted drug design. Right? So next, consider that what is the timeline for drug discovery and drug development. Right? So the drug discovery and drug development is a very lengthy process, and the process may take 14 to 15 years of time. So consider that you have started working in lab and you have discovered some possible drugs. Right? So consider that at a time there are 10,000 drugs that have been discovered. Right? This is the drug discovery step. So suppose we have discovered 10,000 of drugs. So in the next step is the preclinical setup. So in the next step, we consider these 10,000 of compounds, preclinical trials, and of these 10,000 of compounds, only 250 will pass the preclinical trials. So after preclinical trials, these compounds will undergo into clinical trials. So of these 250 compounds, only 5 compounds will pass the clinical trials. After clinical trials, then FDA will review these compounds, and of these 5 compounds, only 1 drug will be approved. So just see the timeline here. So the drug discovery and preclinical steps. So these steps cost you about 6.5 years, and the clinical trials can take about 7 years of time. And for FDA to approve the drug and to make it officially available in the market, it takes about 1.5 years. So you can see it takes about 14 to 15 years of time for a drug to make its way from lab to the market. And see the reduction in size. So you have started with 10,000 compounds and you get only 1 drug in the market after 14 to 15 years of time. Right? Now we have structure-activity relationship. So structure-activity relationship is the relationship between chemical or three-dimensional structure of a molecule and the biological activity. So the basic theme of structure-activity relationship is that the molecules that have similar structure have similar biological function, and the nature and position of the functional group actually determine the biological activity of a drug. Right? So if we change the functional group, if we change the position of the functional group in a drug, then the biological activity will also be changed. So when we are carrying out the structure-activity relationship, what we do is that we carry out systematically structural modifications. Right? So by means of systematic structure modifications, we get different type of lead compounds. So we optimize the lead compounds and we get the drugs. Right? So by means of these structure-activity relationships, by means of these systematic changes, we can change the structure and get different biological activities. Right? So when you consider the structure-activity relationship in a quantitative sense, means that you say that when this change is made, you get this amount of effect, this percentage of effect has been enhanced, this percentage of effect has been reduced, right? So when you consider the structure-activity relationship in a quantitative sense, then this is called QSAR, or quantitative structure-activity relationships, right? Let's consider that you have a chemical compound and you are to carry out the structure-activity relationships. What we do is that firstly we have to screen that compound, right? The screening can be done by random screening can be done by metabolism studies, or it can be done by identifying the side effects of a drug as well. Right? So once you have screened a compound, then you get a lead. Right? So if a compound you are screening for certain biological activity, right? If that compound shows that biological activity, you get a hit. You modify the hit, you get the lead. Now you have got the lead compound, and this lead compound has to be optimized further, right? The refinement of this lead compound is called pruning, right? So you get the pruning done, right? So you refine the lead and you are doing the pruning. After pruning, what you get a structure in which the functional groups are at their optimized position to show the best biological activity, right? The structure that has the idealized functional group at their idealized position with the best possible biological activity, this structure is called pharmacophore. Right? So pharmacophore is the actual structure which is responsible for the biological activity, right? So you can say that the purpose of structure-activity relationship is to discover that pharmacophore, right? So what we do is that we get a structure, we refine it, we make a lead, and we define the lead to get the best possible combination of functional groups and to have those functional groups at the best positions so that we get the best biological activity. Right, so in order to screen for a chemical compound, to identify lead compound, there are different methods of screening. So we can screen by means of random screening, right? So random screening is done when you don't know nothing, when you don't know anything about your receptor target. Then we have non-random screening. For example, we are not getting some compound which is lead compound by any way. So what we do is that, suppose that in random screening we get some hits which are not very suitable hits because they are not showing much better activity, but they are showing some weak activity, right? Those compounds which were showing some weak activity in the random screening, suppose that we have similar compounds to that, right? Suppose we have two compounds, A compound and a B compound. The B compound was showing some weaker interaction, some weaker activity in random screening. A compound has not been tested by random screening, right? But this A compound resembles this B compound. So when we consider this screening of this A compound, this is called non-random screening. So non-random screening also called focused screening, because you are focusing on certain things, right? So random screening is not focusing on the structure, it's just a way to just search for a possible structure because you don't know anything about the receptor, right? So non-random screening is more focused thing. So in case if you have a structure which was not showing better activities, was showing some weaker activity in the random screening, and you have another structure which resembles this structure that was showing some weaker activity, then if we search or if we screen for that structure which was resembling the first one, this is called non-random screening. Then we can identify the metabolites of the drug, see if the metabolites has some better activity or not. Then we identify the observable side effects of the drug, see if the drug has some side effects of the drug that has maximum biological activity with minimum side effect. That will become our lead compounds, right? So this way, by observing the side effects, we can identify a lead compound. So suppose we have the best structure, we have the best pharmacophore. Now the drug can show biological activity only if it is able to bind with the receptor, only if it's bind with the target, right? So in order for the drug to be able to reach to the target, it is essential the drug must be able to get solubilized in the body. It should be able to absorb through the cell membrane. So these two things are very important. The drug should be able to be solubilized in the biological medium, and the drug should be able to cross the cell membrane. It should be able to be absorbed through the cell membrane, right? So there are different factors which are involved in these processes. These include the ionization, solubility, pKa, hydrogen bonding, protein binding, isomerization, chelation, and bioisosterism. So in general, we say that a drug is able to be absorbed only if it is in unionized form. Right? So if you have a molecule which is not ionized, it will be absorbed very rapidly. But if a thing is ionized, then depending on whether it's an acid or a base. So if your drug is acidic in nature, then in that case the drug will be absorbed only in acidic medium. And if your drug is basic in nature, for example, if your drug is an amine, then in that case the drug will be able to be absorbed only in basic medium. The reason is that the acidic drugs remain unionized in acidic medium, while the basic drugs remain unionized in basic medium. Right? The drug must be unionized in order to be absorbed. Second parameter is the solubility. So drug must have some hydrophilic character as well as hydrophobic character. Right? Drug must have polar character as well as non-polar character, because by means of polar character the drug will be able to show affinity for water, because the medium in in blood is water. So in order for the drug to be absorbed or solubilized in the blood, it must have some water, it must have some hydrophilic character. In order to cross through the cell membrane, the drug must have some non-polar character as well, right? The drug must have hydrophilic as well as lipophilic character both. Then we have pKa. So pKa value actually affects the ionization of your acid. Then we have hydrogen bonding. Hydrogen bonding affects the solubility in the water. The hydrogen bonding can be optimized, can be increased or decreased in order to get you the desired solubility. Then we have protein binding. So once the drug is inside your blood, then it has to be carried out to the receptor only once it's bind with the proteins, right? So there are proteins in the plasma. They are called plasma proteins. So what we do is that once the drug is inside your blood, the plasma proteins bind with the drug and carry the drug throughout your body. And once the drug reaches the receptor, the drug is dissociated from the protein and it gets attached to the receptor. Right? So it is important that the plasma binding with the drug should be reversible. Right? But if the drug forms a covalent bond with your protein, then in that case the bonding will be irreversible. In such cases, we get toxicity because of such bindings, right? Then we have isomerization. So we have isomerization, both stereoisomerization, the position isomerization, the conformation isomerization, right? Different isomers have different biological activities. So if you have different conformational isomers, so different conformations have the ability to interact with different receptors, and since different receptors have different biological activities associated with it, so if your drug binds with different receptors in different conformations, then your drug can have a number of side effects as well. So it's important that the number of conformations the drug has should be limited, right? So if your drug has limited number of conformations, it means that it will be able to bind with limited number of receptors, and the number of side effects will be reduced. Then we have stereoisomerism. So stereoisomers are the isomers which differ in their arrangement in three-dimensional space, right? So the stereoisomers can bind with receptors to different extent. As a result, the different enantiomers will show different biological activity, right? Because they differ in their three-dimensional arrangement, therefore they are not able to show same biological activity. So one enantiomer may show greater activity, other may show lesser activity. It's also possible that one enantiomer will show the biological activity while the other will be toxic as well. The next parameter is chelation, right? So chelation is a process in which your drug binds with a metal, right, and forms a cyclic metal complex. It's called a chelate, right? Now there are two situations. Number one is that your drug binds with a metal which is playing important role in the body function, right? So when your drug binds with such metal, in that case the metal will not be able to play its biological role. As a result, we will get some diseased state in the body, right? For example, zinc is an important metal, and zinc is a part of insulin, right? So if your drug binds with the insulin, with the zinc, then in that case the insulin will not be able to work, right? So drug should not bind with a metal which is essentially playing some role in the biological system. The other context is that suppose that you have high metal toxicity inside your body. So if your drug comes and binds with that toxic metal, so this metal drug is helping you to eliminate the toxic metal from your body. So this is a positive concept of chelation, right? So chelation should be such that the drug is able to bind with the heavy metals to remove toxicity, but the drug should not bind with the metals that are playing important role in the body, right? Next is biisosterism. So biisosters are the atoms or group of atoms which have similar physical, chemical and biological properties, right? So suppose you have a drug molecule and you want to modify it. So one way is that just replace a biisoster with another. So by replacing biisoster with another, the biological activity is not lost, but there is chances that you may get rid of some side effects as well, right? So let's consider, we have briefly described all of these parameters. Let's consider that we are taking a drug in dosage form, right? So you are taking a drug, whether it's in the form of tablet or in the form of syrup or whatever. So drug has to be solubilized inside your body. So these factors, which include the ionization, pK, hydrogen bonding and isomerization, so these factors determine whether a drug will be able to get soluble inside your body or not. So once the drug has been solubilized, it comes into your bloodstream. Now the drug has to cross the cell membrane, right? So in order to be absorbed through the cell membrane, these are the parameters which affect absorption. That's partition coefficient, hydrophilicity, lipophilicity, and the ionization, right? So once the drug has been absorbed, next thing is that drug should be able to reach the target, to reach your receptor and bind with it, right? So for that purpose, it's important that your drug binds with a protein, and the binding should be reversible. So these proteins will carry your drug to the receptor, and once the drug reaches the receptor, it will interact with the receptor to show the biological activity. Right. Another term which is related to this process is the maximum absorbable dose. So maximum absorbable dose is the amount of drug that is actually absorbed at a certain dose. For example, I'm taking 1 milligram of a drug. So of this 1 milligram of the drug, the maximum drug that is absorbed by the body is called maximum absorbable dose, right? Some of the drug has to be lost in first pass metabolism or some other process, right? So this maximum absorbable dose, which is absorbed at a certain dose, is called maximum absorbable dose and is abbreviated as MAD. Then we have stereoisomers. So stereoisomers differ in their three-dimensional arrangement in space. Because of their difference in three-dimensional arrangement, they show different biological activities, right? And bioisomers are the groups or atoms that have similar physical and chemical properties. Now we have Eason and Stedman hypothesis. According to this hypothesis, different enantiomers show different biological responses because different enantiomers bind with the receptors differently. According to Eason-Stedman hypothesis, only those drugs will be able to show biological response which can form a 3-point fit. For example, let's consider two enantiomers. So one enantiomer is the isomer 1 and the other is enantiomer is isomer number 2. So consider this one. So this isomer is forming 3-point fit because 3 atoms are complementary to the receptor site. So this flat surface is your receptor site and the balls are the atoms of your enantiomer, right? So the A is complementary to A prime on the receptor, B is able to bind with B prime, C is able to bind with the C prime. So this enantiomer is forming a 3-point fit. So this isomer number 1 will be able to show the biological activity. While the isomer number 2, you can see that C is complementary to C prime, B is complementary to B prime, but D is not complementary to A prime. So this isomer is only forming a 2-point fit. Right? So according to Eason-Stedman hypothesis, the isomers which are able to form a 3-point fit will be able to show biological responses. So if an isomer is able to form more than 3-point fit, the biological activity will be more enhanced. But it is a minimum criterion that for any system to show some biological activity is it then show that your drug must form a 3-point fit with the receptor. Right? So next we have analogs approach. So for example, you are carrying out structure-activity relationships and you are considering the systematic changes. So what systematic changes you can do is that you can make homologs. Homologs means that you can increase or decrease the chain sizes. You can increase or decrease the ring size of a drug molecule. Then you have molecular fragmentation, means you can add or remove certain fragments to see what change in biological activity we get. We can add or remove certain functional groups. We can also change the position of the function groups to see the changes in the biological activity. Then we can carry out bioisostatic replacements. Right? We can change the stereochemistry, right? And we can change the ionization. If a drug is ionized form, we can convert it into unionized form. If a drug is in unionized form, we can see what changes in biological activity we get if the drug is converted into ionized form, right? So let's consider a few examples here. This is example of molecular fragmentation. Means here in cocaine, if we are removing this ester group, right? So we are replacing this ester group with the hydrogen, right? So we have removed this ester group, but we get tropococaine, and the tropococaine is more potent than this cocaine, right? So you can see the removal of the fragment is enhancing the biological activity. Here we have addition of a functional group. So we are considering this tolazolene. So tolazolene has some biological activity, but if we add another benzene ring to this tolazolene, the biological activity is enhanced. Right? So benzene is a functional group. So this is example of addition of a functional group. Right? Similarly, we can remove a functional group or we can change the position of a functional group and see what changes we get in the biological activity. This is example of bioisostatic replacement. So we have uracil and here we have a hydrogen here, right? So when we replace this hydrogen with a fluorine in uracil, so what we get fluorouracil, and this hydrogen and fluorine, since they have similar sizes, similar electronic configuration, so they are bioisosters with each other. So replacing hydrogen with the fluorine, we get some enhanced biological activity. Now we have another example of bioisostatic replacement. Suppose we replace this oxygen with a sulfur, right? So replacing this oxygen with a sulfur enhances the biological activity. This is the example of bioisostatic replacement. Now we have some examples of stereochemistry, right? So we are changing the isomers. So this is one position isomer. If I just change the position of a methyl group and bring it here, like this one, right? So this is another medicine, and this medicine in which the position of this methyl group has been changed, this medicine is more potent than the parent medicine, right? So the pentobarbital was the actual parent medicine, and the amobarbitol was the drug that was formed by changing the stereochemistry of the medicine, right? Then we have ethambutol. Ethambutol has two isomers. It has two enantiomers, right? One enantiomer is this one and the other is that one. So if we have this enantiomer, if we have this stereoisomer, it has anti-tuberculostatic activity. While if I change the stereochemistry of this one, what happens that this isomer causes blindness, right? So changing stereochemistry can have drastic effect on the biological activity. Next we have change in ionization. So we have dimethyl sulfonium analog. So this is ionized state of the drug. So it has some biological activity, but if we convert this charged analog into permanently uncharged sulfide, in that case the biological activity is enhanced, right? So this is the analog approach in structure-activity relationship. I hope you have understood this lecture well. So thanks for